airplane mode.
Okay, we had regulation of gene expression in chapter 18. And if we allow ourselves to picture the opening of an orchestra, there are many sounds going on that come to mind, and we may hear the plucking of strings, the random squeaks of wind instruments, and the occasional clash of a cymbal. But the moment the conductor raises his baton and begins the direction of the instruments, joining and fading out of all the discordant sounds are transformed into a beautiful, properly balanced and timed symphony that captivates the audience. And in this way, cells must similarly and intricately and precisely regulate their gene expression. And so the focus of our presentation is to explore how bacteria regulate expression of their genes in responses to their different environments, how eukaryotes regulate gene expression to maintain different cell types, how gene regulation can allow a single cell to become a fully functioning organism made up of many different cell types, and how disruption in gene regulation can lead to cancer. And a lot of this stuff we already reviewed when Doc went over it. And so just a quick review, does anyone remember what an operon is? Jack?
Section on the RNA strand, It's not some type of strand that activates something.
Well, an operon is a unit of genetic function found in bacteria and phages, consisting of a promoter, an operator, and a coordinatedly regulated cluster of genes whose products function in a common pathway. What about a repressor? Anyone? Tony?
A repressor would stop the creation of RNA.
Yeah, it inhibits transcription. So in prokaryotes, it represses. In prokaryotes, repressors bind to the DNA in or near the promoter. What about a regulatory gene? Cameron?
Is that like a gene that like regulates how much of like something is made?
Well, yeah, it codes for a protein, like a repressor, and it will control the transcription of another gene or a group of genes. And then a co-repressor is a small molecule that binds to a bacterial repressor protein and changes its shape, allowing it to switch the operon off. Now, with that being said, does anyone know what an inducer is? Jack?
Doesn't it bind to a repressor that is usually active on the strand? It binds to that repressor and thus breaking off the repressor allows transcription to go?
Well, it binds to the operator, and then it switches the operon on. So it does the opposite of a core repressor. And then an activator is a protein that binds to a DNA and simulates gene transcription. Jacqueline.
Okay. So I'm going to start with explaining the differential gene expression. So at any given time, only about 20% or a fifth of our protein coding genes, that's excluding introns, are expressed at any single moment. Specialized cells require less expression of this DNA, so such as nerve cells, they don't always express 20% of their protein coding genes because they don't always need it. But every somatic cell, except for the immune system, has the exact same DNA. So the differences in these specialized cell types doesn't come from the content of the DNA, but how it's expressed in the individual cell. So transcription factors are needed to activate the right genes at the right time so that the cell can function the way it's supposed to help the organism survive. And failure of the DNA to be expressed properly can lead to disease and organismal disarray, such as cancer. Essentially, a signal is received that calls for DNA modification, and then leads to transcription, RNA processing, translation, and then the protein degrades. It's kind of like signal transduction, where a cell receives the signal, and then a bunch of pathways are ignited that carry out the signal expression. So advances in DNA technology have allowed for the enhanced understanding of these processes, which we previously didn't know a lot about. Regulation at the stage of transcription is a universal administration point for gene expression in organisms, meaning that every organism can regulate their expression at the transcription stage. So let's review the chromatin structure. The DNA of every eukaryotic cell is enclosed around a protein in a network termed chromatin, which is essentially every, like all the chromosomes wrapped and DNA wrapped around histones, and they're all bound together, and they are condensed in heterochromatin. So this assists in the regulation of gene expression based on the type of chromatin that the DNA is in. So the gene's promoter locus affects whether or not the gene will be articulated, but typically only genes found in euchromatin, which is a less densely packed form of chromatin, is expressed because it's easier to access it because it's not wound as tightly. Chemical alterations to histones within the chromatin can influence configuration as well as expression. So depending on if the histone is methylated or acetylated, it will affect how the gene, or if the gene is expressed. And we'll go through that later. Okay. So histones consist of a body, which is essentially just like a ball, and then they have an end terminus tail, which is just a tail, and that protrudes outwards from the DNA from like the DNA's part that is wrapped around. The tail is composed partially of amino acids, and they are accessible for chemical adjustments such as acetylation. So an acetyl group can be attached to the lysines in the histone tail, and then once acetylated, the lysine's positive charge is neutralized, and the histone tail dissociates binding to neighboring nucleosomes. So essentially, there will be a bunch of histones, and they're all wrapped in tightly together in a heterochromatin. And then once the tails of the histones are acetylated, they become less tightly wound, and they're more spread apart, and then the genes are more accessible for gene expression. Histone modification continued. So ordinarily, the binding between histone tails causes the chromatin to fold more tightly within itself. This is referred to as heterochromatin. And when the binding does not occur, the chromatin is loosened to euchromatin. Transcription processes can access and bind to the acetylated regions more freely. So deacetylation is the opposite of acetylation. It's the taking away of those acetyl groups. And research suggests that histone acetylation enzymes may foster the actuation of transcription by recruiting constituents of transcription apparatus. So essentially, if a histone is acetylated, it almost calls for the other parts that are needed for transcription to start. So it's a recruiter, so to say. Methyl and phosphate groups can fixate and dissociate within the tail's amino acids. So essentially this can bind and then unbind as it's needed. DNA methylation. Just like acetylation,